the way the cancer cells develop either through mutations where you alter the function of certain genes or through silencing of certain genes through epigenetic mutations, alterations. And both of those mechanisms are occurring in colorectal cancer. We know that there are certain types of colorectal cancers where epigenetic modifications play a bigger part. Those tend to be like tumors that originate, for example, in the right side of the colon, tumors that have mutations in a gene called BRAF. Those tumors tend to have more epigenetic mechanisms underpinning them. And again, the way the field has moved in colon cancer over the past maybe 5 to 10 years is that we are more and more and more aware of the fact that colon cancer is not one disease, but it is multiple diseases. And those diseases differ in the way in their etiology and how they come about. And also they differ in how they behave. And they differ in the way we should treat them, although we're not always there at that point. But eventually the treatment would be very different. And those, again, divisions could include the tumors that are driven mainly through epigenetic modulation, like the right-sided tumors, and the tumors that are not driven through epigenetic modulation, or where epigenetic modulation, not a high player, but it's more driven through oncogenic alterations and mutations in key genetic pathways. The interesting thing about epigenetic alterations is that they are very heavily influenced by our lifestyle and our habits. So that's why there is this whole other field that is looking at ways of prevention of colon cancer through alterations in epigenetic modifications and things that can alter epigenetic changes. Of course, smoking, tobacco is a big contributor to epigenetic mutations or alterations. Dietary things like eating certain fruits and vegetables it could expose the colon to natural compounds that have ability to reverse epigenetic changes. And then exercise, people feel now exercise could contribute to that as well. And recently there have been a number of papers that have shown, for example, if people have colon cancer and they're treated with curative intent, if they have a sedentary lifestyle versus a very active lifestyle, the risk of recurrence of the colon cancer is much lower if you have an active lifestyle. In the past, there have been many studies that have looked at relationship of diet, especially eating vegetables and fruits, and lowering the processed meats in the diet. And all of these approaches that impact lifestyle most probably have effects on epigenetic modifications, and that's the way on the biologic level they're causing these alterations. Now, are there tests for this? There are a number of tests that are now available for screening for colon cancer. Some look at the alterations in the stools, such as those tests would rely on detection of alterations in epigenetic changes. And many of the circulating DNA tests where we look for prints of the cancer in the blood, those also rely on measuring epigenetic changes in the circulating DNA in the blood. So alterations in epigenetic changes are commonly used in screening and in monitoring colon cancer these days. So, the CRISPR system, the CRISPR-Cas system, the identification and discovery and modification of that approach has been a huge revolution in medicine. And needless to say that a few years ago, the two scientists who discovered this got the Nobel Prize for that discovery is just a testimony to the importance of this discovery. And this system enables us to edit genes within cells. Of course, on the bench, like if we're doing experiments in the bench side or preclinical work, this has been a huge revolution because it enables us to take out certain genes, insert other genes, whether in the cell line or in animal models, to study the effects of those genes. Now, there is interest also in using this in human disease, and the first applications are in diseases that are driven by one gene, like, for example, sickle cell, where you could use CRISPR to edit the sickle cell gene and then replace the progenitor cells with the healthy gene instead of the mutated gene. So in those diseases that are driven by one gene, this is there. Now, in cancer, there is a huge promise for this technology, but it is a promise at this point. It's not been an applicable treatment yet. And the promises are that we could use CRISPR-Cas to uh, edit the genes that are like mutated. We could also use CRISPR technology to insert or add the genes that are or edit genes that in fact impact the immune response to the tumor. So, for example, we could modulate a gene and then bring in a CAR T against that or bring in an antibody against that. So that could be another approach. And of course, with epigenetic editing, could be another approach where we could use a similar technology rather than to go after a specific gene, but to go after parts of the genome where there is epigenetic changes. And those all are very promising technologies. They, in theory, could alter the way we approach cancer and treat cancer. But again, there are also certain challenges in applying that technology in patients.